Hi, my name is Mary Park, and it is week 3, lab 5, which is the bacterial morphology and staining techniques. Um, so in this video, I've already done the actual experiment itself already. And so I just want to basically show you exactly what is needed to do the experiment. And then in the next video, I'm going to go ahead and show you the results of it. So for this exercise, so it's a simple staining technique, and it's going to be techniques with direct and negative stains and to prepare the smears. You want to be able to identify the basic morphology, including bacteria's arrangement. So with a direct, it's going to be the use of my teeth and my gums. You want to be able to grab the cells, and then you want to put like a dime size of it, and then you want to completely air dry, as well as you need to like light it right past over the candle flame for 5 times, no more than a few seconds. And then you want to let it cool down for 10 minutes, and then you want to put in the sliding tray, and then you want to be able to add 1 to 2 drops of crystal violet stain for at least 60 seconds, and then gently rinse it over water. So let me show you exactly what was done, what I did. Sorry. Okay, so this was the end of result of it, but the materials that is needed. So these are the actual what is need to stain. We have the crystal violet, the iodine, decolorizer, as well as the safranin. So here we go. This is the staining tray right here, the candle needed for aseptic technique, and also to put it the slides over the flame. And again, this is the candle lighter, as well as your goggles, your mask, your gown, and your gloves needed to practice safely when doing any experiment. Okay, so that was for the direct M. And then now for the negative, I would need to do a single drop of Congo Red. However, in my result, I'm going to show you that I had to get some information on the internet, just because the fact that I did not have Congo Red and be able to explain it closely. So again, it's exactly the same thing as the direct. However, what you would need to do, and I'm going to explain, is that you would need to put a single, you would go ahead and collect again, put it on a slide, and then you would need to make sure that you would air dry it, and then yeah, you would need to air dry it for at least 30 minutes, and then, which I will have to show you what it would look like, since I was not able to do that experiment since I didn't have the Congo Red. Alrighty, so differential stains. With differential stains, pretty much you're going to prepare bacterial smears with gram stains using pure samples from broths, which will be inoculated from the previous experiment. Let me show you again exactly what is needed. So these are the cultures that were inoculated from the previous experiment, and these are the slides that are labeled. The E. coli, S. cerevisiae, S. epidermidis. So I would transfer a dime size into the actual slide, and then I would fix heat the sample passing the light, which is on the candle, and then I would put a drop of crystal violet on the actual slide for 60 seconds, and then I would rinse it with water and then add 2 to 3 drops of iodine, wait for 60 seconds, and then gently rinse it off on the staining tray, and then add 2 to 3 drops of decolorizer, wait for 30 seconds, gently rinse it in the staining tray, and then add two to 3 drops on the safranin, and then wait for 30 seconds, and then repeat for each slide, and then at the end I would be able to view it underneath the microscope, which will be the result, which I will show you in the next video.